The entrenched clinical standard for weight management relies on a single metric, a strict daily energy deficit of 500 to 750 kilocalories to dictate fat loss. Most professionals categorize time-restricted eating, or TRE, within this model, viewing it as a behavioral guardrail to prevent late-night snacking and ensure a caloric deficit. This split-screen metabolic flow reveals a clinical anomaly. On the left, 1,000 kilocalories enter the system at 8 a.m. and route smoothly toward muscle and energy output. On the right, the exact same 1,000 kilocalories enter at 10 p.m., resulting in a bottleneck of hepatic lipid storage and blood glucose spikes. Even when caloric loads match perfectly, physiological outcomes diverge based solely on when food is consumed. Chrononutrition is mathematically independent of caloric restriction. This phenomenon challenges the "a calorie is a calorie" heuristic that forms the bedrock of modern dietetics. Strict isocaloric clinical trials are the only empirical method to isolate the variable of time from the variable of energy by ensuring both groups consume the exact same amount of food. If isocaloric trials demonstrate diverging cardiometabolic outcomes, then the foundational clinical model of human metabolism is critically incomplete. Establishing these findings requires the rigorous institutional environments of places like the National Institutes of Health, where every gram of intake is precisely controlled. In the Hattori-Mirin model, subjects were fed an obesogenic, high-fat diet under strict isocaloric control. One group was fed ad libitum, or whenever they chose, while the other was restricted to a specific feeding window. This graph tracks the Hattori outcomes. The time-restricted group was completely protected against high-fat diet-induced hepatic steatosis, or fatty liver disease, and glucose intolerance, despite consuming the identical caloric volume as the control. These results have seen direct human clinical translation, specifically within the context of managing prediabetes. The Sutton prediabetes crossover trial observed men practicing early time-restricted eating, or ETRE, where all meals are consumed in an early 6-hour window. Researchers meticulously calorie-matched all meals against a 12-hour control schedule. The Sutton physiological results showed significant improvements, a 25% increase in insulin sensitivity and a 7 mm of mercury drop in blood pressure for the ETRE group. Most critically, these cardiometabolic improvements occurred with absolutely zero weight loss. By holding weight and calories as constants, these trials prove that circadian alignment directly and independently alters systemic metabolic efficiency. To find the source of the isocaloric paradox, we must shift from external dietary behavior to the internal transcriptional machinery of the cell. Human metabolism is governed by a tightly regulated transcriptional-translational feedback loop of clock and BMAL1 genes, which oscillate daily to coordinate cellular energy production. This system is anchored by a central pacemaker, the suprachiasmatic nucleus, or SCN. Located in the hypothalamus, the SCN is entrained exclusively by light cues. The SCN communicates with a network of peripheral oscillators located in metabolically active tissues, specifically the liver, pancreas, and adipose tissue. These peripheral clocks are primarily entrained by feeding behavior, known as non-photic Zeitgebers, rather than by light. During daytime feeding, the light-entrained SCN and the food-entrained peripheral organs fire in perfect biological synchrony. Consuming calories during the biological night acts as an asynchronous cue, disrupting this coordination. Late feeding forces the liver and pancreas to process nutrients while the central SCN is signaling the body to enter metabolic dormancy. This state is called uncoupling. This physical uncoupling of peripheral clocks from the central pacemaker is the mechanical root cause of chronodisruption and metabolic inflexibility. Beyond genetic oscillators, we can observe this timing in the daily amplitudes of systemic metabolic hormones. Every morning, a sharp surge of cortisol enhances gluconeogenesis, the process of creating glucose from non-carbohydrate sources to mobilize energy. This morning surge correlates with peak pancreatic insulin sensitivity and maximum carbohydrate tolerance. Early feeding leverages this window, resulting in high diet-induced thermogenesis and efficient nutrient oxidation. As the day ends, core body temperature declines, and the body's thermogenic capacity plunges. This evening decline coincides with the onset of nocturnal melatonin secretion. Melatonin actively binds to pancreatic receptors to suppress beta cell responsiveness and halt insulin secretion. Beta cells are the specialized units in the pancreas that produce insulin to lower blood sugar. Carrying the MTNR1B risk allele amplifies this melatonin-induced insulin suppression, a genetic vulnerability present in a large subset of the population. When late-night calories hit a pancreas that has been chemically shut down by melatonin, the result is severe postprandial glucose excursions. Eating late at night is an active endocrinological collision that directly damages glucose tolerance. Timing is the first pillar of this model. The second is the structural sequencing of macronutrients upon intake. Macronutrient preloading involves prioritizing proteins and viscous dietary fibers before consuming carbohydrates. Inside the gut, viscous fibers like beta-glucans inhibit alpha-glucosidase, an enzyme that breaks down complex starches, to blunt immediate glucose absorption. The microbiome then converts these fibers into short-chain fatty acids, or SCFAs. These SCFAs travel to the lower gastrointestinal tract to bind with intestinal L cells. This binding stimulates the L cells to secrete endogenous GLP-1, a hormone managing blood sugar and satiety. This endogenous pathway differs from pharmaceutical GLP-1 agonists by regulating appetite naturally while preventing the loss of lean body mass. Managing the sequence of macronutrients programs the gut to output insulin-sensitizing hormones, acting as a biochemical force multiplier for time-restricted eating. We must shift toward a multi-dimensional clinical framework that integrates circadian biology into the energy balance model. Optimization requires early time-restricted eating, front-loaded morning calories, and consistent protein and fiber preloading. This shift is reflected in high-level medical consensus, with institutions like the American College of Cardiology now integrating these complex interventions into standard care. The isocaloric data proves that circadian alignment restores metabolic flexibility and limits visceral fat independent of standard energy deficits. Chrononutrition functions as an independent, mathematically proven physiological lever required to restore human metabolic homeostasis.